Hello, my name is Caroline Nelson. I am a senior biology student and Markey Star Fellow at UNCG. Here, I will be presenting the research that I have conducted in Dr. Rebecca Sappington's lab at Wake Forest University over the previous summer and current spring semester. Moving into the background, one of the lab's main objectives is to further our current understanding of the role of interleukin-6 in retinal health and vision. Interleukin-6, or IL-6, is a cytokine that has traditionally been characterized as a mediator of inflammation. During immune responses and natural aging processes, the expression of IL-6 is upregulated. Axons of retinal ganglion cells form the optic nerve projection and perform a critical role in transmitting visual stimuli to the brain. Neurodegeneration in the retina is characterized by transport defects, degeneration of synapses and dendrites, and inflammation. In humans, retinal ganglion cells, axons, enter the thalamus to reach the visual cortex. However, in mice, the majority of axons project to a visual processing region of the brain called the superior colliculus. In a paper published last year, the Sappington lab revealed that the cytokine is critical to the development of healthy retinal ganglion cells in young 3-month-old mice. In fact, it was determined that axonal transport of the neural tracer Cholera Toxin Beta from the retina to the superior colliculus was halved in IL-6 ligand knockout mice. The relationship between retinal function and IL-6 is believed to be mediated, in part, through interactions between the microtubule-associated protein stathmin and the major downstream effector of the molecule of IL-6, STAT3. Seen in Figure 1, STAT3 and stathmin proteins interact in healthy retinal ganglion cells to stabilize microtubules and allow for cargo proteins to carry out normal cellular transport processes. When STAT3 and stathmin interactions are disrupted, microtubule organization is not maintained and a deficit in transport occurs. With the aim of better understanding the role of IL-6 signaling in retinal health, we examined the effects of the loss of the cytokine in an aging model. To make this assessment, the following steps were performed. First, it was necessary to identify archived tissue of experimental and control groups. The experimental group was composed of IL-6 knockouts greater than 10 months old, which had been injected with Cholera Toxin Beta, or CTB, at 3 and 5 day time points. After appropriate tissue was selected, optic nerve sectioning and immunohistochemical labeling of STAT3 and stathmin, followed by confocal microscopy, was performed to assess the presence of both proteins in retinal ganglion cell axons. Additionally, sectioning and wide-field microscopy of the superior colliculus was performed to visualize and quantify CTB transport. The injection schematic of Cholera Toxin Beta is illustrated in Figure 2. Here, it is important to note that retinal ganglion cell axons emerging from the right retina project to the left superior colliculus, and axons from the left retina project to the right superior colliculus. So, when red CTB is injected into the vitreous of the right eye, it will be transported to the left superior colliculus and vice versa. From left to right in Figure 3, representative images of sectioning from the beginning, middle, and end of the superior colliculus are displayed in 3A. The left hemisphere of the superior colliculus, fluorescent green in this cohort, displays 5-day transport time point. The right hemisphere, in red, displays the 3-day transport time point. Here, it is visually apparent that CTB transport to the right superior colliculus is lower than that of the left. Figure 3B represents quantification of CTB transport in young and aged IL-6 knockouts. In the young 3-month-old group, a difference in transport is apparent between the 3 and 5-day time point, with the 5-day time point being similar to that of the control. Interestingly, in the aged group, CTB transport is reduced at both time points. In both the young and aged models, 3-day transport of CTB yields a greater fluorescence in the control group than in the IL-6 knockout group. Again, from left and right, Figure 4A depicts the immunohistochemical labeling of aged IL-6 knockout optic nerves. In the 10x and 60x magnifications on the optic nerves, on the left, CTB is red and STAT3 is green. On the right, CTB is green and stathmin is red. Nuclei are labeled blue in all images. It is clear that both proteins are present in aged IL-6 knockout animals. Figure 4B represents heat maps of CTB fluorescence in each group. Heat maps from 3-month-old mice are on the top row, which display a visual representation of CTB transport in the 3-day control and 5 and 3-day knockouts. In the second row, the same is shown for 12-month-old animals. Again, deficits can be observed in the 3-day knockout groups and aged 5-day knockout group. In this presentation on interleukin-6 mediated axonal transport, we have shown that the absence of the cytokine signaling confers reduced transport of the neural tracer Cholera Toxin Beta in aged mice. A previous study by Warham and colleagues described reductions in 3-day CTB transport in young IL-6 knockout mice, as well as aberrant microtubule organization. When compared to controls, aged animals have reduced axonal transport. Here, we have shown that a decrease in axon transport of CTB is present in both 3 and 5-day time points in 12-month-old IL-6 knockouts. From immunohistochemical labeling, we have observed the presence of both STAT3 and stathmin in aged optic nerves. Moving forward, we intend to further assess the relationship between STAT3 and stathmin in aged interleukin-6 knockout animals in several ways. First, we will complete immunohistochemical labeling of aged control group to determine any differences in the presence or localization of STAT3 and stathmin. We also intend to discern any protein-protein colocalization through proximity ligation assays. Thank you for taking the time to watch my presentation. My email is in the description box below should you have any questions.